Hello friends, welcome to our channel, Gajpal A Learning. In this video we are going to see about the topic, pharmaceutical errors and their related topics. So let's start the topic. Pharmaceutical errors. In pharmaceutical analysis, errors refer to discrepancies between the expected and observed results during the testing, measurement, or analysis of pharmaceutical substances. These errors can arise due to various factors, such as instrument inaccuracies, sample contamination, procedural mistakes, and environmental conditions, leading to inaccurate or unreliable data. It's crucial to minimize and account for errors to ensure the quality and reliability of pharmaceutical products. Classification of errors. Errors in pharmaceutical analysis are typically classified into three main categories: systematic errors, random errors, and gross errors. Let's delve into each category in detail. First, systematic errors or determinate errors. These errors are consistent and occur in the same direction each time measurements are taken. They are often caused by flaws in the measurement setup, calibration issues, or improper techniques. Systematic errors lead to results that are consistently either higher or lower than the true value. They can be difficult to detect but can be minimized through proper calibration, standardization, and careful experimental design. Second, gross errors or blunders. Gross errors are significant mistakes that occur due to human errors, equipment malfunctions, or improper techniques. They can lead to results that are far from the expected values and are often easy to identify upon review. Gross errors can be caused by things like misreading instruments, incorrect sample preparation, or transcription errors. To prevent gross errors, careful attention to detail, cross-checking results, and following standard operating procedures are essential. Third, errors in measurement. Errors in measurement refer to inaccuracies that occur when determining a quantity. These can be a combination of both systematic and random errors. Proper measurement techniques, calibrated instruments, and controlled experimental conditions help reduce errors in measurement. Uncertainty, which quantifies the range within which the true value lies, is associated with every measurement and is often used to express the degree of error. And other errors. Other types of errors can include parallax errors, caused by an incorrect viewing angle when taking measurements, interpolation errors, inaccuracies when estimating values between data points, and instrumental errors associated with the limitations of measurement instruments. Additionally, operator errors, such as incorrect pipetting or mishandling of samples, can introduce errors into the analysis process. Next is sources of errors. Certainly, here are some common sources of errors in various processes, including pharmaceutical analysis. First, instrumentation and equipment. This involves calibration issues. Instruments that are not properly calibrated can lead to inaccurate measurements. Drift. Instruments that change their readings over time due to factors like temperature fluctuations. Non-linearity. Instruments that don't provide a linear response across the measurement range. Second, sample handling and preparation. This involves contamination, introduction of foreign substances during sampling or preparation, incomplete mixing, uneven distribution of components in a sample, evaporation, loss of volatile components during sample preparation. Third, environmental factors. Under this, temperature and humidity variations, these can affect measurements and reactions differently. Light exposure. Sensitive samples can degrade if exposed to light. Airborne particles, dust and contaminants in the air can interfere with samples. Fourth, human factors. Under this, procedural errors, mistakes during experimental steps or procedures. Observer bias, the tendency of an analyst to see what they expect to see rather than what is actually present. Fatigue and concentration, human limitations that can affect accuracy. Fifth, chemical reactions. Under this, side reactions, unintended chemical reactions occurring during analysis. Decomposition, breakdown of compounds due to exposure to heat or light. Sixth, sampling techniques. Under this, sampling bias, selecting non-representative portions of a sample for analysis. Sample heterogeneity, inhomogeneity of samples leading to inconsistent results. Seventh, measurement techniques. Under this, parallax errors, incorrect reading of measurements due to viewing angle. Interpolation errors, estimating values between data points inaccurately. Resolution limitations, instruments with limited precision can round measurements. 8. Data recording and analysis. This involves Transcription errors, mistakes when transferring data from instruments to records. Calculation errors, errors in performing calculations during data analysis. Inaccurate software, faulty software or algorithms used for analysis. 9th. External interference. This involve electromagnetic interference, signals from electronic devices affecting measurements. Cross-contamination, transfer of substances from one sample to another. 10th. Standardization and reference materials. Under this, poorly prepared standards, standards that aren't accurately prepared or calibrated. Inaccurate reference materials, materials used for comparison that have their own errors. Next is method of minimizing errors. Certainly, here are several methods for minimizing errors in various processes, including pharmaceutical analysis. First, calibration and standardization. Regularly calibrate instruments to ensure accurate measurements. Use certified reference materials for calibration and validation. Establish standardized protocols for calibration procedures. Second, quality control measures. Implement quality control checks at various stages of analysis. Include replicate measurements to assess precision and consistency. Monitor and maintain the performance of instruments over time. Third, proper training and documentation. Train personnel in accurate measurement techniques and protocols. Maintain detailed and accurate records of all procedures and measurements. Fourth, sample handling and preparation. Follow standardized procedures for sample collection and preparation. Minimize exposure to external contaminants during sample handling. Fifth, environmental control. Maintain consistent temperature and humidity conditions in the laboratory. Shield samples from light exposure if they are sensitive to light. Sixth, data entry and analysis. Use electronic data recording systems to minimize transcription errors. Implement double-checking mechanisms for critical data. 7th, instrument maintenance. Regularly service and maintain instruments to prevent drift and inaccuracies. Implement a preventive maintenance schedule. 8th, cross-validation. Compare results obtained from different methods or instruments to identify inconsistencies. Use different techniques to analyze the same sample as a form of validation. 9th, use of automation. Use automated instruments and robotic systems to reduce human error. Automate repetitive tasks to improve consistency and accuracy. 10th, appropriate experimental design. Ensure that experiments are well planned and controlled. Incorporate proper controls and reference standards. 11th, adherence to standard operating procedures, SOPs. Develop and follow standardized protocols for all processes. Regularly update SOPs to reflect best practices and improvements. 12th, peer review and collaboration. Have colleagues review your work to identify potential errors. Collaborate with others to benefit from diverse perspectives. 13th, continuous improvement. Regularly assess your laboratory processes for potential sources of error. Implement changes and improvements based on lessons learned. 14th, statistical analysis. Use statistical tools to analyze data, identify outliers, and assess measurement variability. Apply appropriate statistical methods to quantify uncertainty. Last topic is accuracy and precision. Certainly, accuracy and precision are two important concepts in measurement that describe different aspects of how close a set of measurements are to the true value and to each other. First, accuracy. Accuracy refers to how close a measured value is to the true or accepted value. In other words, accuracy indicates the absence of systematic errors. A measurement is accurate if it is very close to the actual value, regardless of whether the measurements are consistently higher or lower than the true value. High accuracy means that the average of the measurements is close to the true value, even if individual measurements might deviate. Second, precision. Precision refers to the degree of reproducibility or consistency among multiple measurements of the same quantity. In other words, precision describes the ability to obtain the same result repeatedly under the same conditions. High precision means that the measurements are very close to each other, even if they are not necessarily close to the true value. It's possible to have precise measurements that are consistently off from the true value. Example: accuracy and precision. Suppose the true concentration of the active ingredient in the drug is 100 mg per tablet. First, accurate and precise. Under this, measurement 1, 99 mg. Measurement 2, 100 mg. Measurement 3, 101 mg. In this case, all three measurements are close to the true value of 100 mg, accurate, and they are also very close to each other, precise. Second, accurate but not precise. Under this, measurement 1, 97 mg. Measurement 2, 100 mg. Measurement 3, 103 mg. While these measurements are accurate because they are not far from the true value of 100 mg, they are not very close to each other, not precise. Third, precise but not accurate. Under this, measurement 1, 92 mg. Measurement 2, 95 mg. Measurement 3, 98 mg. These measurements are consistent with each other, precise, but are consistently lower than the true value, not accurate. Fourth, not accurate and not precise. Under this, measurement 1, 85 mg. Measurement 2, 105 mg. Measurement 3, 90 mg. These measurements are both far from the true value, not accurate, and widely dispersed, not precise. Thanks for watching this video. If you like the video then please do subscribe to our channel and support us.